Yeah, thank you, Margot. Thank you, Alberto. Thank you, audience, for coming. It's a remarkable place, Milano. Milano can be very proud of Leonardo da Vinci, who famously depicted the Vitruvian Man. And the Vitruvian Man depicts the perfect physical dimensions of the human body, the perfect proportions. And for hundreds of years, we didn't know the perfect time dimension. What is the perfect age of an organ in a perfectly healthy person? And nowadays, we do have ways of measuring the ages of all human organs, all human cells that contain DNA, based on chemical modifications of the DNA molecule. And these chemical modifications of the DNA can perhaps be interpreted like rust accumulating with time. And so as we age, there are certain gains of methylation, gains of these epigenetic changes, and similarly loss of methylation at other parts of the DNA. And one can measure time based on that. So today I will talk about these epigenetic clocks that measure age. I could measure any of your ages. You could come to me and say, Steve, here's a blood sample, or I could use your saliva. I could take a tiny bit of your skin, of your fat. So any source, I could take a cup of your urine. You pee in a cup, I can measure your age. Why? The urine contains a bit of DNA. And so these epigenetic clocks measure aging based on these DNA samples. My entire talk is about DNA methylation, which is a change to one of the letters of the DNA. The DNA contains 4 letters, and one of them is the letter C. And so cytosine methylation changes during development and also aging. The purpose of methylation and epigenetics is to control which genes are turned on and which genes are turned off. And this epigenetics is crucially important during development. A zygote could never develop into an embryo without methylation. But also we now know that methylation plays a role in the aging process. So here I show you an epigenetic clock curve. Please look at the blue curve. As we age, methylation age increases quickly during development, but after age 20 or so, there's a linear relationship. So then there's a linear change with aging. And epigenetic clocks are defined as prediction methods. It's fundamentally done in statistics, machine learning, artificial intelligence, so quantitative science, computational biology, bioinformatics. So this field has various statistical approaches to predict your chronologic age, how old you are according to your passport, but also methylation can be used to predict time to death. How many years are left until you die? So it predicts your mortality risk. I will talk a lot about a particular epigenetic clock, which is known as Grim age. Grim age is named after the so-called Grim Reaper, Father Death, so Grim Reaper. This it's a methylation clock. It looks at 1,000 locations of the DNA, 1,030 locations. Some of them gain methylation, others lose methylation. And this Grim age measures your mortality risk. So let's say I take a blood sample from a 50-year-old, and if the Grimm age is 58, then this person has 2 times the risk of dying the next year than a regular 50-year-old, the average 50-year-old. So we published recently so-called Grimm age version 2, and again it predicts time to death. And how do we do that? We typically use blood samples collected maybe 30, 40 years ago, and for each person we know, are they still alive, or if they died, when did they die, what did they die of? And therefore this is known as a retrospective study. So using then these blood samples from the 1990s, we can build predictors of time to death. And Grimm age is quite interpretable. Although it is a clock based on methylation alone, we can interpret these methylation changes in a variety of ways. Some of the methylation changes relate to your smoking history. If you smoke a lot, Grimm age is greatly accelerated. Other methylation changes relate to particular proteins in the blood that are informative. For example, there's a protein called growth differentiation factor 15 that's elevated when people experience mitochondrial dysfunction or senescence. And also it measures some methylation changes correlate with markers of inflammation, such as C-reactive protein. Others relate to glucose levels, hemoglobin A1C. So Grimm age is strictly a methylation clock, but it does relate to inflammation, smoking, and mitochondrial dysfunction, glucose disruptions, and so on. So Grimm age offers several advancements compared to previous epigenetic clocks. It has the best predictive power of mortality in our comparative analyses. It can also predict time to coronary heart disease and time to physical decline. It relates strongly to smoking patterns. And it also, it mainly applies to blood, but some people have used it successfully in saliva samples. I want to give you a visual demonstration of the predictive power of Grimm age. So here you see different patterns that show survival curves. Here and in different epidemiological cohorts. For example, in the upper right panel, you see the Chianti cohort, which is a famous cohort in Italy. And we looked at blood samples collected in the Chianti cohort, and we compared the 20% fastest agers to the 20% slowest agers according to Grimm age. And you see a good separation of the survival curves. So the fast agers, you see the red curve, within 15 years, many of these people unfortunately had died. But the slow agers, according to Grimm age, the blue curve showed a much better pattern. And here I show you many panels, all from different cohorts. As I mentioned, Italian cohorts, American cohorts, European cohorts, Scottish cohorts, all stratified by sex. And so Grimm age very much predicts mortality risk in any group that an epidemiologist could define. For example, it would predict mortality risk in older women who are heavy smokers. It would still predict mortality risk. Or younger women who never smoked, or African Americans, or so different racial groups, So it really predicts mortality in these different groups, which demonstrates that it has information beyond smoking, beyond traditional risk factors. So GrimAge has been consistently shown to be the most accurate predictor of mortality and morbidity. There are many hundreds of publications have evaluated it. And I will now describe several applications of GrimAge in different groups. And I start here with an Irish cohort. So here, epigenetic clocks were applied to blood samples from an Irish group. And I want to draw your attention to the evaluation of exercise in the middle. So you have here low exercise, moderate, high, and so on. And here's GrimAge acceleration. A negative value indicates that these people age more slowly. And what do you learn? Interestingly, these people who self-reported physical exercise, their GrimAge was a little bit reduced. Conversely, here you see smoking, huge effects. That didn't surprise me. So yes, if you smoke, then yes, GrimAge is greatly accelerated. So this was an Irish study. Let's now go to a different population. These are now Taiwanese people. So here, an independent group of researchers evaluated blood samples from 2,500 Taiwanese participants, and they wanted to see whether epigenetic clocks relate to cardiovascular health scores. What are those? Health scores, your smoking status, your physical activity, what do you eat, body mass index, are you obese, cholesterol, blood pressure, glucose. So researchers can measure your cardiovascular health score. And in this Taiwanese study, GrimAge and other mortality predictors were, again, as expected, related to cardiovascular health. Now I come to a completely different cohort. These are no longer Taiwanese people. This is now an American cohort, the so-called CARDIA study, a famous study for evaluating cardiovascular risk factors. And this was a very nice study, 20 years of follow-up of these participants. And every year, the researchers evaluated many biomarkers and assessments of cardiovascular health, obesity, blood pressure, cholesterol, glucose, smoking, exercise, every year. But in year 15 and in year 20, they actually added a methylation clock test, a blood sample from that time. And then they asked, well, did GrimAge reflect cardiovascular health changes? And over 20 years. And the answer is yes, you know. So, but there was an interesting interpretation. So I'm quoting here from the paper. Our findings suggest that GrimAge acceleration may be a useful biomarker of early stage cardiovascular disease before it is detectable using current technologies. So it's a bold statement. And that's kind of the dream of methylation clocks, that the dream is that we measure methylation clocks frequently, maybe once a year, maybe every 2 years, and it could be informative of health changes before other biomarkers pick them up. I'm not saying we're there yet. I phrased it as a dream, you know, but you see there are encouraging data. I now want to come to a completely different cohort. We now go from the US to Germany. There was a study in Germany in the Rhineland by an independent team, and they evaluated 3,600 German participants in a cross-sectional study. And they measured physical activity using something called an accelerometer. fundamentally, they don't trust what you tell them, whether you exercise. They want to see it. So as you know, there are many devices for measuring physical activity. It could be your iPhone or a smartwatch or Fitbit. So you can measure physical activity. And here in these people, again, interestingly, physical activity measured by step count, how many steps you walk, was again significantly associated with lower grim age. And I think, to me, this is somewhat of a miracle, to tell you the truth. It's stunning that when you exercise, somehow it changes your DNA. That's really what the insight is. Remember, a methylation clock measures changes to your DNA. So somehow that has an effect, But the study did more. So we know physical activity affects, let's say, grim age, but why? What mediates these changes? So in that study, they looked at cardiovascular factors, and they found that blood pressure, adiposity, meaning your obesity level, and triglyceride levels mediated the effect of physical activity on epigenetic or grim age. And what that shows us is that grim age kind of reflects your past behaviors, Now we come to yet another completely different group. We now move from Germany to Hungary. So here Zsolt Radak did a quite interesting study recently. So he looked at 80 volunteers, and these were not average people. These were serious athletes. They were rowers, participants of the Olympia, very fit people. And in this study, he measured epigenetics, but excitingly also the microbiome. So you can microbiome, how? It takes a stool sample and sequences the stool and looks at bacteria in your stool. And I was very interested in this study because I always wanted to know whether the microbiome, your stool, relates to epigenetic age. And so he had to study 80 people. And now I first looked at the stool microbiome data, and I was, to be honest, shocked. By training, I'm a statistician, so I look at data in a certain way. The first thing I ask, is there information in the data? Start with that. So I look at the microbiome data, and the first observation is males are completely different from females, which is, why would that be? It makes no sense to me, but that was the finding. And also, there were a few women, and I hope you can see it if you have a good eye. There's some red, do you see these red bars? So there is a couple of women looked completely different from all the other women. And again, I have no explanation whatsoever why that is, So for me, the first impression of these microbiome data was that these are very challenging data, differences between males and females. There are even strong differences within groups of women, But anyways, the next thing we looked at is whether the gut microbiome data correlate with age. Do they change as we age? And if I'm honest to myself, I would say there's barely a signal in it, So in this group of people, and to remind you, these were between 38 and 84, If you want to flatter older people, you call it middle-aged people. So, but in that group, the microbiome barely changed with age, So. But in any event, but whatever changes we saw were again sex-specific, And also the relationship between the microbiome and epigenetic age, if there was a change, it was sex-specific, So unfortunately, in my opinion, it will be challenging to combine these two hallmarks of aging. Hallmark of aging is epigenetics, and the other hallmark of change is the microbiome. I think it will be difficult to reconcile those. So epigenetic, I want to summarize the epigenetic clocks. So epigenetic clocks will, in my opinion, be valuable for identifying and even validating anti-aging rejuvenations. However, when you just want to predict mortality risk or lifespan, there are many alternatives. You can measure blood pressure, smoking, history, frailty indices, lipids, glucose, and so on. So, and all of these predict lifespan. But interestingly, epigenetic clocks seem to enhance these standard lifespan predictors. And for me, the great advantage of epigenetic clocks is that they must relate to at least one root cause of aging, a primary cause why we age. And what do I mean by that? What I mean by that is, imagine you have a person who follows the perfect lifestyle. They sleep 8 hours a day, they exercise an hour a day, they have an easy job, no stress, they eat vegetables, they do everything right. Guess what? They still age and they still die. Why? Well, there are these root causes of aging, and my hope was always that methylation clocks relate to at least one of these root causes. By the way, I don't know the time. 2 minutes. Okay. Okay. Yeah, so a couple of days ago, somebody talked about clones, and I want to mention something. As we get older, some of us accumulate mutations in stem cells. For example, blood stem cells, hematopoietic stem cells. These are so-called cancer-causing somatic mutations. And there were a couple of papers that found that mutations in these methylation-altering enzymes do relate to epigenetic age acceleration. So this condition, this clonal hematopoiesis of indeterminate potential, these clones, these cancer clones, are strongly associated with epigenetic age acceleration. So I thought it was interesting. I want to end by showing you the future of comparative biology. A few months ago, there was a major paper in Nature Medicine on the validation of biomarkers of aging. And here the consortium said there's a particular type of validation that's very important, which is called cross-species validation. Because if you can validate something across different animal species, then it's more likely to be connected with aging as a universal phenomenon. What does that mean? We know every single mammalian species ages. We know dogs age faster. We know whales age more slowly, but they still age. All mammals age. And would it not be nice to have a biomarker that measures aging in all mammalian species? And to address that great challenge, I organized the Mammalian Methylation Consortium from researchers around the world, 200 people, and we built these multi-species clocks. And the trick is we predict the relative age of an animal. And the relative age is defined by the true calendar age of the animal divided by the maximum lifespan. It's a ratio. The relative age of a 2-year-old rat is 0.5. Why? Rats can live up to 4 years. So 2 years divided by 4 is 0.5. And the relative age of a 61-year-old person is also 0.5. Why? Because the oldest person who ever lived, Jean Calment, lived until age 122. So in any event, so multi-species clocks predict this relative age based on cytosine methylation. And so here I show you on our pan-mammalian clock, this is now one regression model, one set of highly conserved cytosines on the DNA, and this clock measures relative age in all mammalian species. Here I show you the pan-mammalian clock in the upper left applied to humans. It measures human relative age. It measures mouse relative age, dolphin, elephants, bats. So it works in all species. I want to, final comment, we can also predict the maximum lifespan of a species based on methylation. We analyzed 348 different mammalian species, and if you give me the DNA of a species, I can predict the maximum lifespan, the gestation time, and so on. And I'll stop with that. Thank you so much.